Good afternoon to everybody. So people, I'm here specifically today. My whole purpose with this lesson is to prepare you for the 7th of May. Now, for learners who are viewing this online, we are here in the Free State. You are viewing across the country. You're also writing similar assignments, so you can benefit from this discussion as well. But in Free State Province, learners will be writing a formal assessment on the 7th of May on genetics, and I've got the scope which I've handed out to the learners here in person. And for those who are not here, they can download it. You just go to monetlabproject.co.za, you go to past Saturday lessons, and then by tonight, we'll have this video lesson there, plus the notes, and then people can download it. So those who are not in Free State Province, including our sign language learners, they can download it from the website. But our learners here have the official scope, and I want to run through it. Now please, learners, make a note that for every assignment, every test, every exam this whole year, you will always get general scientific skills that you need to know. What are we talking about? Look at your scope. You need to be able to draw all kinds of graphs. Like, can you draw a pie chart? Let's hear from you. Can you? Yes. Some people forgot how to do it. Just go practice it, because every now and then in life sciences, they surprise us with a pie chart. And when you do a pie chart, please write down there, show all calculations. They give marks for the calculations, how you calculate the degrees of those certain sectors of the big pie chart. You must show the calculations, and always remember heading of a graph and draw the correct type of graph. You know the difference between a bar graph and a histogram, all those you need to know. Now please, on your scope, right there at the second bullet point, the word always. For each and every test, each and every practical, they will ask you what is the independent variable and what is the dependent variable in a certain experiment you've never seen. Independent, please write there, cause, and dependent, write there, the results. So you must go and find what is the cause in a specific experiment or investigation, and then you write down specifically what that cause is, and then the results are the dependent variables. Now, there's a difference in the third bullet point between when you do a certain investigation in science, like let's say you do your thesis for your master's degree, they will always ask, is this investigation valid and is it reliable? Please write there next to it, there's a difference. You need to know what's the difference. Many learners think reliability and validity is the same thing. When we do our theses, like for PhD, they give two different stamps. There's a stamp for reliability of the investigation, and there's a stamp for validity. Go and make sure, even for the 7th of May, you need to know what is the difference. Now, they like to ask about planning steps. Planning steps are always done, obviously, before you set up the experiment. Now, if they ask you about planning steps, I want you to write down, even here on the scope, 3 things that you can always write to score marks when they ask you about planning steps. Please always write, you need to establish and get together the equipment. What equipment will you need to do this investigation? And then get the equipment together. So the equipment, then the next one that you always write is, how will you collect the data and sort the data that you will be collecting? That's a second planning step, so you plan that before you actually get into the investigation. And then if there's permission required, maybe you need to ask permission of parents because you're working with underage children. If there's permission, you must always ask for permission. That is the third thing that you can always write with all planning steps for all investigations. And then, what do they mean? Please raise the hand with that next bullet point. They say, even for the 7th of May, you need to be able to draw a conclusion. Please raise the hand. What do we mean by that when we say, draw a conclusion? Sorry, you want to answer, say again? And then? Well, what does it mean if I say, please draw a conclusion? No, but I think you understand. But let's just hear somebody else. They will always say at the end, now draw a conclusion. They simply mean write down a sentence. You're not going to draw anything, eh? Write down a sentence where you explain what do you now realize from your hypothesis that you set out in the beginning. Hypothesis is a prediction. I predict something like the longer the winter is, the colder the temperatures are going to be going into the winter. That's my prediction now. Now I will gather the temperatures over the next 3 months, and then I will say, I reject my hypothesis, because what I predicted is not actually what happened. But you must then specify what really did happen. Then you write there. Now here's your conclusion. As we proceeded with winter, the temperatures actually increased, and that is contrary to what I predicted initially. See, so you write out what is your findings at the end, and you come to a conclusion. You see, conclusion means the end. What do you actually learn from this whole investigation? Then they will give you simple calculations to do on the 7th of May. One of it is percentage increase. Now, all of us know how to calculate the percentage of something. If you score, let's say, 27 out of 30 for the practical assessment on the 7th of May, what's your percentage? But they will be asking on the 7th of May, calculate the percentage increase of something. Let me use an example. Let's say we have here on today's date. You can write this example, you can write it down just as an example. On today's date, on this area, this premises here on campus, we've got of a certain species, we've got this number of birds, of certain species of birds, certain kind. That's a number of birds we have. By 31 December, we have, let's say, this number, they have increased because they laid eggs and they had chicks, so they increased in number. Now they ask you, in this particular kind of bird, what is the percentage increase in this year, of this calendar year? Then you always use this formula, please. For any percentage increase, you always write the starting point at the bottom, and you write the difference on top. And you multiply it by 100 because we want percentage. People, you need to know this for the end of the year, but you also need to know it for the 7th of May. How to calculate percentage increase. So what is the starting point here? Where do we start? We're starting today. So the starting point is, that number is 1250. And what is the difference between the two numbers? You must subtract the one number from the other. That means difference. So what is the difference? If you quickly calculate that, you'll get to what? Who can quickly do the calculation? If you calculate, what's the difference between this number and that number? You subtract. It's 580. So you always write the difference. That means the difference in number, how they've increased in number from the starting point going forward. So the difference on top, starting point at the bottom, times 100 over 1. And then you will say, if you do the calculation, there's a 46.4% increase in this bird population. That's percentage increase. Now, how to calculate percentage decrease? If they ask you to calculate, let's say on the 7th of May, percentage decrease of something. Let's say we had here on the premises, let's write this side. Let's say we start off today on the 3rd of May. Of a certain kind of tree species, we had 15 of these trees. We come here at the end of the year and they want to know what is the percentage decrease in that particular species of trees that we have here on campus. Now during the year, some maybe died and some were chopped down, and maybe at the end of the year we count there's 11 trees left of that kind of tree. Could be pine trees or whatever they are. Now you need to calculate the percentage decrease. You use the very same formula. Please help me. What do you see that side? You must put at the bottom the what? The starting point. And what must you put at the top? And you must always multiply by what? Because you're working towards percentage, isn't it? Always when you work with percentage, you must multiply by 100. So, what was the starting point where we said we took account for the first time this year? The starting point is 15. And what's the difference between 15 and 11? 4. Multiply by 100. Now, if you do this calculation, who can do that calculation quickly for us on a calculator? You get to a certain percentage decrease now. but you use the very same formula. Did you say 26? All right, then often we will round it off. So it's a 27% decrease in the tree population. People, are you together? So the way the markers give marks, they normally give a mark for doing this correctly, times 100, and then a mark for the answer. So you can get 3 marks for this whenever you get this. But please make a note of a video that I recorded that I want you to go and view on monyetlaproject.co.za. You go to Saturday school lessons, you go to past lessons, and you go down, you scroll down until last year. On the 16th of March, 2024, I recorded a lesson on scientific investigation skills for a whole hour, that you can watch that lesson to also prepare you for the 7th of May. Please write down the date. It says here, there's a video that you can watch. You can watch the 16th of March, 2024 lesson, not this year, last year's lesson. There's also notes I gave last year that you can download from there with regards to general skills when we talk about scientific investigation skills. But we want to focus our attention now on genetics. There's terminology that you always need to know. You see the terminology here in the scope that you need to take note of. Those are basic terminologies. Now, when we deal with genetics, we especially need to be quick to identify when are we dealing with complete dominance, when are we dealing with incomplete dominance. So you read a question and you say to yourself, okay, now I'm dealing with incomplete dominance. And then the one you also need to know for the 7th of May is co-dominance. So when am I dealing with this? So I need to be on the lookout for something in the question paper. I need to look for something that will tell me I'm dealing with a particular dominance in nature and genetics, and then I need to implement the correct method for it. So, how do we look for it? Please read the definition of complete dominance. Then you raise your hand as soon as you have an answer and give advice to the friends. What should they look out for in a particular question to know that they're dealing with complete dominance, not incomplete or co-dominance? Complete dominance, can you quickly check there? What must learners look out for? In a question, you can also summarize like I'm summarizing it here. What must they look for? Yes. Here, the ladies in front here are saying they're correct. They say you must look out in the question for the word dominant, because that's what it tells you. There is dominance here. Now, we'll talk now about what dominance really means. It's complete dominance. You look out for the word dominant allele. And the opposite of that is what? Recessive. Recessive allele. You look out for these words. Now, the method we use, let me use the example of pea plants. I'm going to use this example here of what Mendel did. The grandfather of genetics, Gregor Mendel, said with peas, you get, if you look at their seeds, have you ever seen some seeds that we eat of peas are yellow in color? Others are green in color. And he said that in peas, he's seen, just like with dogs, black hair color is dominant over brown. With people, brown eye color is dominant over blue. There's dominance in nature. Now, what did Mendel mean? Because it's the truth about genetics. What did he mean by capital letter and small letter? Can somebody please raise the hand? Because that is now dominance. Complete dominance. When there's a capital letter and a small letter, you're going to use it as such. You're going to use capital letter and you're going to use the small letter. You're not going to use G for green. You're going to use the small letter. But what does it really mean? Can somebody just summarize for us again? Raise the hand, make it clear. I know many of you know. What does it really mean? It simply means if you inherit one allele, which is now a variety of a gene, let's say the pea plant inherits from the one parent, the capital Y, and that's how genetics works. You always inherit one allele from the one parent and the other one from the other parent. Now you have this combination. And when you have capital letter, small letter, you call it homozygous or what? Heterozygous. Well done. Heterozygous. You call it heterozygous. Now, Mendel said, if you have the dominant allele present, the capital letter, it dominates the small one in the phenotype. For instance, a certain person can carry the small b for blue eyes in her genes. But if she has the capital B for brown eyes, you will see when I look at her eyes, it will be brown eyes. Because the dominant allele dominates the recessive one. In the phenotype is what you physically see. What is the word we use for these codes we write? What is the word we use? These are the codes we write as a genotype. What you physically see. If I see you've got brown eyes, that's a phenotype. So what will be the color of the seeds of these peas here? Yellow. Not something in between. Not like another color or some. It just will be either yellow or green. When will it be green? You agree with me, it will be like this. Then it will be green. Two small letters. Because the moment the capital letter is there, it dominates. So it can be like this, or it can be two capital Y's, then it will be yellow in color, the seeds. But the moment there's only small letters, then it can be the recessive trait that comes to the fore. Now people, let's say we cross two individuals like this that are heterozygous. There's a certain format which I've emphasized here at our Saturday program that you must use, please, on the 7th of May and also going towards the mid-year exams, September and November. You always use this format. I'm saying, here's the question. We're now going to cross two of these individuals that are heterozygous as parents. So you say, well, this is the way to write it. P1 are the parents. I must write P1. Then I say P1, I summarize it for the marker, the phenotype, and I must summarize for the marker, the genotype. Where do I get this information from? It's in the question paper. I'm not using my intelligence. I'm just using the question paper. Then I'm going to do a Punnett square. That's what you always use. And you draw the 4 blocks like this. And what word do you write here? You write here gametes, the sex cells, gametes. And what forms the gametes, what process? And when these gametes are going to get together, you write here fertilization. And this is the F1 generation. We're working from the parents to the first children. The second children become F2. But we're now working towards the first children. So now we're ready to write. The phenotype, I'm working with P's. What did I say? This parent would look like what? What color? Yellow seed color. So normally you must write in full, yellow seed color. Don't just write yellow. But I'm running out of time. So you write yellow is crossed with, because I said you cross two of these that are the same. Both heterozygous. Then the genotype, I get it from the question paper. You must write it according to this format. Phenotype, genotype. Then I'm ready to put it in my Punnett square. There's a line that divides capital Y from small y. Capital Y, small y. I've just taken this one, I've written it here, I've taken that one, I've written it there. Now you're ready to combine. Bring that down, bring this across. Capital, capital. What's the phenotype of this one? Yellow. What is the phenotype of this one? What's the phenotype of this one? So you just bring the capital Y like that, and this one is? Green. So you see how easy genetics is. People, let me share with you a truth of life. There's nothing difficult in life. Anything that seems difficult is not really complicated. It's just difficult because it's new. That's it. Genetics is really easy. You'll see today. There's nothing difficult about it. You must just become familiar with it by asking questions, practicing, and all of us can do it. It's not something difficult. It's just new to many of you. Then you summarize here. Summarize for the marker. The phenotype of the F1. What can they be? Yellow, yellow, yellow. It can be yellow, or it can be green. Then I count in the Punnett square. How many? 3:1. Then the genotypes. What can be the genotype of these? It can be that option, look there, or it can be that option, or it can be that one. There's no other options there. What is the genotypic ratio? How many look like this? 1:2. How many look like that? And 1 like that. They can ask you to calculate if these two parents are crossed. And by the way, plants, we take pollen from the one plant to the flower of the other. That's how we pollinate it. There's no mating like sperm cells and ova. It's pollen that carries the sperm cells. But anyway, if they ask you, if you have this crossing, what percentage will be green? Percentage green in F1. Then you say out of 4, there's 1 out of 4 times 100 is equal to 25% green chance in the F1. It calculates probability. That's what it does. So if you see in a question and you read about there's dominant and there's recessive alleles, there's capital letter and small letter, then you know you're busy dealing with complete dominance. But how will you know that you are dealing with incomplete dominance? You will simply look for a blend. Example of that is where you have, let's say, red roses are crossed with white roses, and you get something in between. You get pink roses, which will be, there is no dominance. Incomplete means there is no dominance. Now please read on the page that I've given you the types of dominance. Incomplete means there is no dominance. Hence, you will find an offspring that looks different from the parents. The one parent is red flower, other one white, and the children are pink. So what is the proper word? Can you shout out that they use there in the definition when we talk about incomplete dominance? They don't talk about a mixture like black and white gives gray. I call it a blend, and I'm not wrong to call it a blend, but what's a nice word they use there? Well done. They say it's an intermediate form. The phenotype of the F1, the children, is something in between. You don't get like here, yellow offspring and green offspring. No. You get maybe an orange color. When you see that blend, you must know you're just going to use capital letters. Something like this, RR, WW, and then you're going to get the pink ones here in the F1, RW. All of them will be pink because you just use capital letters. There's no room here for small letters because there's no recessive allele. People, did you capture what I said now? No room for small letters here. Now, how do you know that you're dealing with co-dominance? Both phenotypes. Now, both phenotypes are in the children. What do I mean by that? You must look out for patches. Like, let's say, a brown bull and a white cow gives a calf that's born white with brown patches. Not brown and white gives, let's say, gray, no. It gives patches. The moment you see that both, please read the definition on the front page I've given you there. When we talk about co-dominance, both alleles are shown in the offspring. Like black dog and white dog gives a dog with black and white patches. Now, what's the method for that? Once again, you use capitals only. Because once again, there's no recessiveness here. Let's say B for black and W for white. No small letters. You just do it like that, okay? And you always follow the same format. The way the markers give marks, we say, right, the learner wrote P1 and F1. We give 1 mark if you've written both. Some learners forget, then they lose that mark. You must write both P1 and F1. Then you must write both these words. Then we give a mark as markers. We give a mark if you write the phenotype correctly, the genotypes correctly, if you've done the gametes correctly here, and if you've done the Punnett square correctly. And then we even go on to give another 3 marks here for the calculation. But you do it exactly like this in the format for all genetic problems. I can see on the 7th of May, there's a lot of success coming for you. All right. People, now I want you to turn this page, the front page, and quickly with your friend in 1 minute spot the kind of dominance you see there. This was a question I got from a similar task learners did in 2019. Now quickly sit with your friend and discuss what dominance is there shown in A, B, and C, and tell your friend why. Why do you say a certain dominance? All right, let's do it together. Please, there you see the top one, there's some flowers. They can give you the names of flowers that you've never seen. Some flowers that look like this are called snapdragons. You don't need to know what they're called. All you need to look out for is red flowers across with white, and you get that all the F1 are pink. Shout out together. What did you write? What kind of dominance is this? You are all correct. Well done. Then number B. You see there's, I don't know, it's a dark, let's say it's a black cow there. No, the cow is the darker one at the bottom. And then the bull is there at the top. So you get one with patches. So you're dealing with what? Co-dominance. Because then you must be able to write, people, this, please write down there, I want you to go practice from now on until the end of the year. Practice how to explain things in full sentences. Please write it down. Because I don't have time now to dictate to you what you should write here exactly. But why, if they said here in this year, 2019, learners had to identify B with co-dominance, you would have scored the mark. But how would you phrase it by using words? You can do it because you're all good with English. You can do it. So you must always write with complete sentences as much as possible. Why do you call this co-dominance? I would say something like, and I don't want you to copy my words now, but just to give you an example. This is co-dominance because both the alleles here are equally dominant for these two colors. They are both expressed in the F1. Hence, there's patches, and this is called co-dominance. You see, you say as much as you can. People, have you ever heard some people, when they tell a story, they don't get to the point quickly. They speak a lot in making a point. Now, you must become like that. You mustn't be like me. I'm a kind of person, I just want to make the point. You must speak a lot. You must tell the whole story, and that's where your marks come from, okay? And then look at the bottom one. Number C. Number A was incomplete. Number B was co-dominance. But number C is what? Now, can somebody raise their hand and tell us, why did you say number C is complete dominance? Because you see the dominant allele is indicated there with capital Y, and the recessive allele is indicated with a small y. Do you see it there? So there's dominance and recessiveness. Do you see the parent on top are two small y's? What percentage of the F1 are yellow? Do you see it's 50%? So you always look at the Punnett square, it gives you information. Now, people, I want you to look at the pedigree diagram on the next page. This was the similar test you're writing on the 7th of May, but in the year 2021. Now, I'm not sure if they will give you a pedigree diagram, but this is understand the pedigree diagram. Look at Umpo, and is it, what's that person's name there? Norley. Getting married, and you see the key here. Umpo is the male here. And then, how many children are born for them? 2, but John gets married to an outside family, somebody, Tandeka, from another family. And then those 2 had their own children again. Please write down. They will ask you to write the genotypes of all these people. And for the 7th of May, do not use X and Y, please. You do not use X and Y. Because X and Y is reserved for special conditions, which I will discuss with you next week here. On 7th of May, you do not, please, capital letters not, use X and Y. Because X and Y you can't use just because it's a male and a female. You'll go wrong. It's not optional. You use it only in cases of hemophilia and color blindness, and where the disorder is carried on pair number 23. But we're not into that for the 7th of May. Now, here's my point. They could ask you to write the genotypes of all these individuals, or a similar question on the 7th of May. Now, please, what I said last week, you look at the children to know what the genotype of the parents are. Please write it down. Look at the phenotype of the children to know what the genotypes of the parents are. I'll use an example. Let's look at Bongo. Now, you tell me, please, Bongo's phenotype. Here, it's all about eye color. Bongo happens to be a male. You see the square there? With what color eyes? Now, they use capital B and small b. Small b stands for what? Well, what does it say in the question? If you read the question 1 there on the top. In this case, people, you must always read the question. Brown eye color is dominant over blue, they say. Hence, we use capital B, like I said here, and small b. Now, I want to know that individual, Bongo, if you look into his eyes, he will have what colors? Because it says here at the bottom, he's got blue eyes. The key tells you. But my question is, write down there the genotype of Bongo for eye color. Who of you, please raise the hands, who of you say for Bongo it must be like this? Raise the hands. You are correct, eh? Because the moment the capital B is there, Bongo would have had brown eyes. But he doesn't. It's got blue eyes, you can see it. So you must have the 2 small letters. But what does that teach us about John? That's my point. I say to you, you must write down. Look at the children's features to understand the genotype of the parents. Now, John is a parent there. Tandeka, we know, can only be what for eye color genotype? So what is Tandeka's genotype? Small b's. Small b's, nothing else. But don't you think John could be perhaps, because John, does he have brown eyes? He could be like that, or he could be heterozygous like that. But we look at his child. A child that's born from him, from John, is Bongo. And Bongo is 2 small b's. So, where do the small b's come from? We know the mother, Tandeka, could only contribute a small b. And that's how genetics works. 1 comes from the one side, and 1 will come from the other side. And this is where Bongo came about with blue eyes. It could happen that capital B could unite with small b, yes. Then this other child, like there, Mandy, or Mandi, as we would say in Xhosa. Maybe it's Mandisa. Did you see Mandisa there, or Mandi? In English they say Mandy. Do you see she's got brown eyes, eh? That would be when the capital B this side get together with a small b, because Tandeka can only give small b's. But the fact that John himself produced Bongo, then you know John can only be like that, nothing else. Because he's got brown eyes, but he also has the ability to give the small b to his child. He's got brown eyes, but his child can have blue eyes. We understand it, eh? Please always look at that. Now, I want you to understand something about codominance. When you talk about codominance, you look at blood groups. And we spoke about blood groups last week. You can go check the video we recorded here last week. Blood groups is an example of multiple alleles. What do we mean by multiple alleles when we talk about blood groups? Multiple means more than two. Like if you look at the alleles here, for P's it can be capital Y or small y. But for blood types, we write the alleles for blood type A. The way we write it, we write a capital I with the A on top there. For B, we write like that. But there's also for O, an allele that we write with small I. But we said between A and B, there is no dominance. In fact, it is what we say codominance. So if you have the one parent giving in the sex cell in the gamete this allele, and the parent carries the other allele O, the parent will be blood type A because A dominates O. When you feel like writing O, you always write small letter I. Are we together? Now this parent gets married to this other parent, and they're going to have children. So what could happen? This one will meet up. You can put it in a Punnett square, or you can do it like this, and this child will be born with blood type AB. But you see here, this child can also be born, an I can be contributed from that side and an I from that side. There's a chance that this parent with blood type A getting married to B, and the child is born blood group O. Now you've put it in a Punnett square just for you to understand. If we would put it in a Punnett square, it becomes very simple to calculate probability. Because that's what we want to do with a Punnett square. We want to calculate probability. So we take this one parent, here's the genotype of the one parent, and we put it in here, just like we always do. There's the one parent's genotype. Then we take the other parent, given to us in the question paper, and we write it here. And we say, the question tells us, calculate the probability percentage-wise to have a child with blood group O. Then you say, to do probability, you must do a Punnett square. So you say, right, a child can be born with this blood group here. This blood group in the phenotype is AB. This one is blood group what? Please shout out. This child is blood group what? B, because B dominates O in the children here. This blood group will be blood type A. And this child can be born, the I goes across, the I comes down with blood group what? This one is O. So out of 4, how many are O? It's 1 out of 4 times 100 over 1 is a 25% chance if these two people had children, these two, that the child will have blood type O. Are we together? Right, people, now we need to understand something else, and that is a dihybrid cross. You need to know that it's very easy. Don't let anybody tell you it's difficult. Now, can somebody please remind us, what do we mean by a dihybrid cross? Quickly think about it, because you need to know this for the 7th of May. What do we mean? Because we've been doing monohybrid crosses all the time. A cross is when you bring two plants or animals together and they mate. You normally make a cross there. So what do we mean by monohybrid? We've done mono means we look at one thing at a time. Like let's say we looked here at the color of the seeds of peas. We just looked at the color. We did not look at the shape of the seeds. All right. So what do we mean by dihybrid? We look at two things at the same time. Like, look at this question that I've given you here of May 2019. It's called page 3 here. on your notes, it says question 2. A certain family they investigated a certain family to check for 2 things at the same time. Quickly look at it, then you raise your hand, and then you share with us. They were looking at what and what at the same time, not just at one thing. They were looking at hair color and eye color. 2 things at the same time. Now, before we come back to this example here, let's quickly look at what Mendel looked at. Mendel said, let's look at pea plants, because he experimented with peas. Mendel said that in pea plants, tall pea plants, that allele is dominant over short. So up to today, we still use these letters like that. And they will tell you in the question paper, use capital T and small t. That means that this plant is what in the phenotype? Tall. People, are you with me? This plant is tall, because the capital T dominates the small t. It's tall. But so we looked at, what would we call this? Can we say we're dealing here with what? We're dealing with the height of the plant, okay. But then he also looked at the color of the seeds. And he said that at the same time, we can look at the color of the seeds. He says that Y, capital Y, stands for yellow. Like here, previously I just looked at the seed color. Now, Mendel said, let's look also at another feature, not only the seed color, but the height of the plant at the same time. And he says, let's use small y for green. Then he said, let's do a test. Let's cross 2 heterozygous individuals and see what happens. So he says for tallness, they're heterozygous, and for seed color, they're heterozygous. We put it together, no problem. And this side, the other parent, we do the same thing. So now how will we go about it when we cross? Now please let me remind you, when you do a dihybrid cross and you see we're busy with 2 features at the same time, like this question here, quickly look at the question again. We're looking at what and what? Hair color and eye color. Here we're looking at the height, and at the same time we're looking at the color of the seeds. Seed color. Seed color. So there's no problem. What is the phenotype of this plant? It's a tall plant with seeds that are? This plant is tall with seeds that are? So if I have one like this, you tell me, if I have a plant like this, what will be the phenotype of it? And green. You see, you understand it so well. If I have a plant like this, what will be the phenotype? Short and yellow. So there's no difficulty for you to spot that. Right now, when we work with dihybrid crosses, you draw a Punnett square with 16 blocks, not 4 blocks. So it's 1, 2, 3, 4. And then we're going to write here. What do we always write here? People, this word is very important. And look now what I'm going to fill in here. I'm writing gametes there. And what am I going to fill in? So now, please write down. In each block here on the top must be one of a kind of a letter. If there's a T, there must also be a Y with it. Okay. Not just previously, like we said, like in other cases, like here, where we only had 1 letter in a block. Because it's dihybrid, we're going to put 2 letters. So let's start, how do you start with any story? You start at the very beginning. You start at the beginning, you say, right, I see a T, and I'm going to take it with the first Y. Because there must be a T and a Y written here. So I say I'm going to write capital T, and the first Y I can get hold of there is there. But there's another Y there. So this time I'm going to write this combination. I'm going to stick to the first T, and this time I'm going to take the other Y, which happens to be the small one. So then I do that, you see. Now I'm going to take the small t with the first Y. So I just work it from a side, and I say the small t now with the first Y. And then what's left for me is the small t with a small y. That means the second with a second. And I write it like that. Are we together? So there's a rhythm to it. People, it sounds like mathematics. Remember, mathematics and music are related. It works like this. Look here what I've done. All of you concentrate. The first letter with the first of the other kind. Then the first letter this side with the second of the other kind. Then I take the second letter with the first and the second with the second. You see there? So I've written now this parent's genotypes. I've written the gamete combination. Now when they ask you in the exams, write the gamete combinations, they mean this. This is what they mean. Now please, can all of you work together with your friend? Take this parent here and fill it in for us this side, please. You will agree with me? Right. Now, what we need to do now is simply go and combine what we have here. We take this one down like we always do, we take this one across. What do we put together? The two capital T's. Now we put the Y's together. And if you have to say, what's the phenotype of this plant? Tall and yellow. Let's write this one here. Quickly help me to see that last word I made, the little circle there. That one will be capital T what? I bring the small t down, eh? I bring the capital T from there. I bring the small t down. The capital Y. So what will be the phenotype of this individual? Tall and yellow. Let's try this one here. Let's fill it in together. Capital T, small t. Capital Y. What is the phenotype? All right, let's try this one here in this block. So we take, what do we take across? Please help me. And from the top there. So it's two small t's. And then the Y's. Big letter Y and? What is the phenotype of this plant? Short and yellow. Okay. Then let's do this last one here. What should I write in terms of T's? Did I do it correctly? What should I write in terms of Y's? So this one is what color? Short and green. Now do you agree with me that I've got 4 combinations here? If I have to summarize the F1 phenotype, then I should be able to write out here that some of these are what? Some definitely will be tall and yellow, isn't it? Some will be tall and green, if I fill in all the blocks. Some will be yellow and tall, and some, green. Some will be green and tall, and some will be green and short. So you've got 4 combinations. You can have 4 options here, that tall and yellow, tall and green, short and yellow, short and green. So then you must go and count in these blocks. That's what they want you to write at the end. A phenotypic ratio. They want you to go and count how many are tall and yellow. And I've done this one. Then I counted, when I filled in all the blocks, I counted. The ratio here was 9 of them were actually tall and yellow. 3 of them were tall and green. 3 of them were short and yellow. And 1 was short and green. I check my answer if it's correct. If it adds up, always please check. If it adds up to 16, then your answer is correct. What I normally do, I take another color pen for myself, or a pencil which I can erase in the exam again, and I always write here, okay, this one is tall and yellow. And so I fill in, this one is short and green. And then I can make my count easily. Now this example I've done here is very much like the one you see here on page 3. Let's quickly read this. We've got about 5 minutes, and this is valuable time for you because you're going to get this on the 7th of May. something very similar to what I see here at 2.1 on page 3 of the year May 2019. An investigation was done to determine the possibility of eye color and hair color in a certain family. The parents were both heterozygous. So now they want us to write down what will be the genotypes of the parents, but that's very easy because they want to know, they tell us they're both heterozygous. Let's just quickly summarize the question here. The question says that what stands for brown eyes? Let's take brown hair first. Capital B stands for what color? Brown hair. Small b stands for what? Is that really so that brown is dominant over blonde? Among humans, yes. Capital B, brown hair dominates blonde hair. Right, and then for eye color, so this is hair color. Let's now look at the eye color. They use capital G for brown eyes. And they use small g for green eyes. Now it becomes very easy because they say in the first question, write down the genotype of the parents for 2 marks. It's so easy because they say both parents in the question paper, they were what? Please highlight the word there, heterozygous. They were heterozygous. So the one parent was like this for hair color and for eye color. It's heterozygous, isn't it? Hetero means capital small. And they say both parents are like that. Now, how were the questions asked that year? Let's say you were matric in that year. 2.1.2. If a male gamete, now we're dealing with gametes. The gametes are 2 letters only together. Containing the alleles B and G, so they say, now with the gametes, if I would put this in a Punnett square, and I would get there to the gametes, and I have my blocks there. And they say the one gamete was capital B, small b. You see in the question paper they say it's like that. Do you see that? They say if a male gamete containing the alleles, look here, B and G. If it would combine with what? With a female gamete with the same alleles, then they say give the following of the child. So they say on this side the other gamete is like this. Now if they would combine, you put the B's together and you put the G's together. So there's your answer for 2A. Because they say, if these were the parents and they both had these gametes, what will the child be like? The child will have, then in the phenotype, what hair? Blonde hair with what eyes? Brown eyes. Because they said, we must use this and this. That's what the question paper tells me to use. So what's the phenotype of the hair? Number B, blonde, and the eyes will be? Now you've scored 3 marks. Now look at 2.1.3. Give the phenotypic ratio. Now I must go and count. I must now then do for this crossing here. I must put it in a Punnett square. You can go do it at home, and then you count. What will be the ratio? Please do that for yourself as a practice activity. But look at 2.1.4. How many of the possible 16 in the above crossing will have the same genotypes of the parents? So please raise your hand, give advice. What should the learners look out for in a block here? They must find what genotype? And then they must go and count out of the 16 blocks. They must look out for, what does the question say? So what should I write? What should they look out for? They should look out for what? So it looks the same as the parents? That's what the question says. If I do the 16 blocks, go and do it at home and count how many do you find that look just like the parents in the genotype. That was the task given to these learners that year. I'm wishing you all the best for 7th of May. See you next week.